quite a novel discussion, discoveries, and along with other groups as well. And then she also made a great contribution to asthma with the involvement of smooth muscle cells, and now I think that she's trying to look into 3D structures involving smooth muscle cells, neurons, and also now I think it's probably T cells and other inflammatory cells as well. And today we would like to hear from her about this dopaminergic nerves involved in asthma. And take it away, Xingbin.
Thank you, Jianwen. I'm very humbled by being nominated as an established investigator for this very wonderful community. Jianwen, to be honest with you, this first thought when I learned I was nominated is that I'm getting old. I feel like established is equivalent to old. But anyhow, at the same time, I also felt the responsibility of duties. But so far, this community has been extremely supportive of junior faculties, and I have enjoyed tremendously the previous talks given by junior faculties and then new investigators. So that's it. I will start my talk. As Jianwen said, I'm going to talk about nerves in the lung and how the nerves may contribute to the susceptibility of asthma in children. So I've actually gave a talk. Let me see why I cannot. Oh, here. Sorry. I need to somehow cannot forward. Just a second. I cannot forward the.
Xingbin, I think it's because you are using the pointer.
Oh, okay. So if
you can click the arrow.
This one.
Yeah.
Oh, gotcha. Yeah, I see. So I see. Thank you, Yawen. So I actually was invited to give a talk to CALA last year in February 2022. At that time, I talked a little bit about basal stem cells and how the basal stem cells can be modulated by disease. So that's another part of my lab who is working on airway basal stem cells and how airway basal stem cell properties can be modulated by different kinds of diseases. So I just give you guys an alert. We have a new paper coming out in Blue Journal on the May 1st issue of the Blue Journal, describing a very unique way of deriving airway basal stem cells from infants. So this methodology really facilitates the discovery of airway epithelial defects in a number of congenital diseases. In this particular paper, we described epithelial defects in a disease that is very common but very rarely understood, which is congenital diaphragmatic hernia. But I decided for my second time at this community, I'm going to talk about the other half of my lab, which is working on nerve development as well as the contribution to allergic asthma. So I was trained as a neurobiologist from Case Western. Before I joined BU as a junior faculty, the truth is I never actually touched the lung before. I still was wondering why they recruited me there. But anyhow, I decided after I became the assistant professor, I'm going to change all my direction. I go back to my neuroscience. So that's how I started my lab. Although the nervous system is known to innervate the lung, the neural anatomy actually is known for many, many years. Even in the old anatomy book published in 1940s, you already can see these wonderful neurocircuitry, how the neurocircuitry is connected to each other and how it reached to the lung to innervate smooth muscle as well as neuroendocrine cells. However, when I started my lab, nothing was known about how this neurocircuitry was built during development. So just to give you some brief overview of how this neurocircuitry looks like. So what happens is the nerve in the lung is not coming from the neurons who are physically in the lung. So basically, the nerve is coming from neurons located outside, with the neural cell body located outside of the lung. So it's very different from GI. GI has a lot of intrinsic neurons, but the lung doesn't. So the sensory neurons that innervate the respiratory tract actually located predominantly in the petrosal ganglion. These are the sensory ganglion that innervates the airway. And then there's a group of neurons then project to the brainstem. I think Xingbin lab has mapped a unique population of brainstem neurons that receive the sensory neuron input. And ultimately, they send back the cholinergic efferent nerve that travel along the vagus nerve, then go back to the lung. So for many of you who may not think about nervous system on a daily basis, just give you an idea how the nerves look like. So here is a mouse staining. We usually do thicker sections, like 25 micron sections, so you can see the whole track of the nerve rather than just a cross section of a very thin nerve fiber. So in this particular staining, we use the CGRP to label sensory nerve as well as neuroendocrine cells. And then the TOBP3 is a pan-neuronal marker that label all type of nerves. So what you can see here is there is it's a nerve travel along the basement membrane. This is probably the nerve that make contact with the smooth muscle to regulate the smooth muscle contractility. And then you can see another branch of nerve coming into endocrine bodies and make very close contact with neuroendocrine cells. So we believe this is one of very, very important regulation of neuroendocrine cell activity in the lung. So compared to the mouse lung, we believe human lungs are actually much heavily innervated. This is a paraffin section, very thin section. As a result, what you can see here is very, very thin, very, very dot-like cross-section of the nerves. You cannot see this type of long nerve tracts. So I also already told you the two major targets in the airway that are innervated are neuroendocrine cells as well as the smooth muscle cells. And then there are three major types of nerves in the lung. One is the sensory nerve, the other one is the sympathetic nerve, which I'm going to spend most of my time talking about. And then last type is the parasympathetic nerve. This is the nerve that provides acetylcholine to regulate the smooth muscle contractility. So as I said, when I started my lab, little was known about how this entire neural circuitry was built during development. So we spent first 3 years of my career just to look for potential mechanisms. So what we did is expression analysis for all the known neurogenic factors during development, as well as genetic models that are deficient in one or two of these neurogenic factors. So very, very quickly, we narrowed down to one neurogenic family members, one neurogenic factor family called the nerve growth factor family. So there are 4 members, but only 3 members of this family are expressed during development in the lung. These are nerve growth factor, brain-derived neurotrophic factor, and the neurotrophic number 4. So what this neurogenic factor is doing is they are produced locally by the cells that are going to be innervated. For example, the neuroendocrine cells make neurotrophin 4, smooth muscle cells make BDNF as well as NGF. So these locally made neurotrophins then get secreted and create this very nice chemical gradient to attract us, so the neurons can know where to send the axon into. So once the axon reaches the lung, the neurons that express the right receptor will contact this, and then this neurogenic factor will feed on these neurons to support the survival of the neurons as well as the maintenance of this axon outgrowth. Eventually, network of nerves that innervate the airway smooth muscle cells as well as the neuroendocrine cells. So if you don't have any of these 3 neurotrophic factors, what you would see here is a much reduced density of innervation as well as fewer axons. So this is a whole left lobe staining with this pan-neuronal marker TOBP3. This is staining at E14.5. So you can see here, if you don't have BDNF, you have much reduced number of axons as well as the branches towards the tip of the buds. The one thing that we learned very fast is the neurogenesis actually follow beautifully the airway branching morphogenesis. Partly is guided by sonic hedgehog regulation of BDNF expression. So once the sonic hedgehog regulates the smooth muscle maturation, the smooth muscle cells start to make BDNF and NGF. Then the nerves coming along. So this is a very beautifully timed as well as spatially regulated process. So after we learned all these important neurotrophic factors, we were wondering whether these factors may play a role in airway diseases. So very quickly, we ran into a whole body of literature talking about the nerve growth family members, how they contribute to asthma. So this is just
can I ask a question?
sure.
So you know that neuron blood vessels are important for morphogenesis in other tissues, I think.
Right.
I know that in adult, if you cut out neurons, it actually doesn't affect tissue very much, lung tissue very much in our hands. But in development, if you, for example, your BDNF mutants,
Yeah.
do you see that the effect coming from neurons?
So if you, so BDNF, none of the neurotrophic factor by itself affect lung branching morphogenesis. So there's one paper published last year from UCSF group. They show that if you do not have BDNF and NT4 all together, the lung is much smaller, and then alveologenesis seems to be compromised. We had that finding as well, but the important thing is these neurogenic factors are so important for brain development. So if you don't have BDNF or NT4, the entire mouse embryo is just very, very, very poorly developed. So, and also give you some ideas, BDNF is also known to be very important for the diaphragm innervation. So you could imagine there's many other confounding factors that may contribute to lung developmental defects without these very important neurogenic factors. So I wasn't convinced to some extent the BDNF and NT4 or NGF controls the nerve development directly, but I'm convinced it may have an indirect effect on lung development.
So I think it's, if you want to design experiments to test, can you somehow, for example, inject a certain neuron toxin or something like that into pregnant mouse, pregnant mother, and in somehow that we hope that this can go into the embryo and kill those neurons. Is it possible to have a certain, because I agree with you that BDNF we have this systematic effects and indirect effects.
Yeah. So the best experiment in my view, so for example, you would imagine you can use smooth muscle actin-3 and then cross with BDNF, for example, but that also ruined the vasculature development. We tried that, so it's not good enough. So that the only way I can think of is, for example, if you have a neurotransmitter receptor that's highly specific expressed in type 2 cells, for example. So then that can really guide you to understand what this nerve is doing. I felt if you're just trying to mess up the entire nerve system in the embryo, I don't really know how can you do that. You might be, maybe with the larger animal, you might be able to inject directly into amniotic fluid because the amniotic fluid is connected directly to the lung. But with the small animals, it's really difficult. I see what you are saying, Jianwen.
Yeah. Okay. Well, thank you. Wonderful.
Yes. Yeah, but I think postnatally, if we can create a very type 2 specific or smooth muscle specific neurotransmitter receptor deficiency animal, then that would be wonderful.
Yeah. Okay. Well, that's a good way to think about it, maybe.
Yeah. I think the whole body or even smooth muscle conditional, neuroendocrine conditional, still not good enough because, as I said, these neurotrophic factors are so important for pretty much every single peripheral organ development.
Okay.
Yeah, but a good question. So based on this whole body of literature talking about neurogenic factor expression is deregulated with allergic asthma, for example, the BDNF expression in asthma is positively associated with asthma severity. So we decided to take a look at asthma just to understand what these neurotrophic factors are doing, and most importantly, what the nerves are doing in the lung in disease. So I probably don't need to make too much introduction about allergic asthma as one of the major chronic respiratory diseases. But I do want to emphasize a couple of things. The first one is asthma usually starts in childhood and then progresses in the adulthood. This is the first thing I want to emphasize. The other thing I want to emphasize is currently the treatment of asthma is only targets the symptoms. The symptoms include type 2 inflammation, the airway constriction. None of the treatment actually have benefit over long term of the slow down the disease or prevent the disease from progression. So as a result, there is no cure. So part of the problem of asthma field, in my view, is all the animal models people built to study asthma is adult model. So this failed to capture the ontogeny of allergic asthma in early childhood. The most importantly, if you started with adult, we believe that many things are already different in adults, not only immune system. Most importantly, the adult lung is also already matured. It doesn't really reflect potentially the developmental environment when the asthma is starting. So because of that, we decided to create a new model. This model take into account the asthma always happens in young children. So what we did is very, very simple. Rather than sensitize and challenge adult animal, we use postnatal day 5 animal, postnatal day 10 animal to start the sensitization, followed by 5 doses of challenges, the same as the usual adult model. The reason why we use this is P21, if you read the literature, comparatively, P21 lung in mouse is almost equivalent to 3 to 5 years old in children. So 3 and 5 years is the first time period you can actually diagnose asthma. So as a result, we decided to take this model. Basically, it's the same as adult model. The only difference is allergen sensitization and challenge happens during postnatal developmental time window. And then we created 2 different models. One is the ovalbumin model, another one is house dust mite model. So they have the same results. So what I'm going to talk about is mostly focusing on the house dust mite model. So the striking differences between these 2 models is the neuroplasticity. So if you compare directly how the nerve level looks like, so this is a Western blot. Blotting for neurofilament is a specific component of the nerves that innervate the respiratory tract. What you would appreciate here is neonatal allergen exposure significantly elevate the amount of neurofilament in the neonatal mouse lungs, but the adult allergen model do not. So we have published a number of papers talking about different aspects of the nervous system in this childhood asthma model. We published a whole series of papers describing how nerve plasticity can deregulate airway smooth muscle contractility. So these are the series of papers we published. We also studied how this neuroplasticity may deregulate neuroendocrine cell secretion that contributes to mucus production in the airway epithelium. But today I'm going to focus on the topic we're very interested in because this contributes to the chronic asthma, which has to do with type 2 cell inflammation. So Th2 stands for T helper 2 cell. So this is the major cell type that contributes to allergic asthma by producing type 2 cytokines. So the material I'm going to talk today is I will briefly summarize the work we published in 2019 on immunity. Most of the data I'm going to present is currently in press by Jackie. So I forget to mention these 2 wonderful people. The entire Th2 project is led by 2 very talented people. Wei is currently an instructor at MGH. He joined my lab about 6 years ago. Jonathan is a research assistant, but he is a very, very impressive immunologist. So he's currently a medical MD Ph.D. student at the University of North Carolina. So I will start the research part by dividing my talk into 2 parts. The first one has to do with how the early nervous system development during this postnatal time window contributes to type 2 cell differentiation. So the type 2 cells has to come from naive T cells. So basically what happens is if you have allergen, the allergen will be presented to the naive T cells, and the naive T cells has to be instructed by a number of cues as well as cell interactions, and eventually they will become Th2 cells. So the Th2 cells make IL-4, IL-5, and IL-13. They also express the key specification factor called GATA3. So these are the drivers for all the phenotypes you can see in allergic asthma attack. So basically you have neutrophil, you have eosinophil infiltration, you have mucus overproduction, as well as have hyperreactivity of the smooth muscle. So this is the first part of my talk. And then the second part of my talk has to do with how these early life exposure can be progressed into adults to become persistent asthma. So this has to do with how the memory Th2 cells are guided and formed during this very critical maturation time window between the initial exposure to become fully functional lung. So this is the two parts of my talk. So the part one of my talk, as I said, has been published. So I will just give you a very quick summary because this builds the foundation for our follow-up study on how these memory cells are formed. So as I said, we initially focused on the embryonic lung environment and how the lung becomes innervated during embryonic stage. So when we started the asthma project, we decided to take a look at postnatal lung development as well as the nervous system development. As we all know, the lung continues to mature and develop postnatally. So the first experiment we did is actually relatively simple. We decided just to look for different types of nerve markers to see how they change during the first 3 weeks of postnatal life. So what we found here is there is little change in cholinergic innervation. This cholinergic marker is cholinergic receptor, one of the cholinergic transporters. So you can see here from postnatal day 5 all the way to postnatal day 21 at adults, there's relatively little change in terms of the content of this cholinergic marker. If you also look at the sensory innervation marker, CGRP, we also see very little change over the postnatal time period. But the only marker that changed very dynamically is the sympathetic nerve innervation. Here we use two different markers. One is tyrosine hydroxylase. The other one is dopamine beta hydroxylase. I will tell you what these two enzymes functions are during the biosynthesis of neurotransmitters for sympathetic nerves. So what we found here is very striking time postnatal developmentally regulated expression of these two markers. The tyrosine hydroxylase increases during postnatal development, and then they're followed by a very sharp decrease in adults. So here is the measurement of the TH levels. So, and then if you look at the DBH, what happened is the DBH continues to climb up, and then the level actually reaches the highest in the adults. So what is the TH and what is the DBH? So I have to show you here. So TH and the DBH are the key enzymes for dopamine biosynthesis as well as the dopamine degradation. So tyrosine hydroxylase is the key enzyme that converts tyrosine to dopamine. And then dopamine beta hydroxylase is the key enzyme that converts dopamine to epinephrine and the non-epinephrine. It is known in the adult lung, the sympathetic nerve is predominantly generating non-epinephrine and epinephrine. So they call it adrenergic nerves. So what happens in this developmental time window, we believe, is initially the sympathetic nerve likely are dopaminergic because they predominantly are TH positive. But with time, the dopamine slowly converted to epinephrine and non-epinephrine because you have a higher level of dopamine beta hydroxylase. So to confirm that, we did the simple ELISA. We grinded up the lung and we measured the neurotransmitter content. And consistent with our prediction, there is a very high level of dopamine at the postnatal day 21, and then this level decreases significantly in adults. Non-epinephrine is consistent with the literature. The non-epinephrine level is much higher in adult lung than in the neonatal lung. So with that in mind, we decided to take a look, where is dopamine being made? Is dopamine made by other cells rather than nerves? So based on the staining here, staining is for tyrosine hydroxylase. As I said, it is the rate-limiting enzyme for dopamine biosynthesis. TUBB3 is the pan-neuronal marker. What we did is we calculated proportionally how much TUBB3 area is taken by TH positive area as a measure of TH innervation. So you can see here, with age, we also see a similar reduction in a proportion of nerves that are Th positive. We also did a similar work with a human lung, and we found in children here is between 1 and 13 years old. These are we quantified as children. And you can see the relative content of Th positive nerves is also much higher in children than in the adults. So when we found these phenotypes, we also ran into literature that's published already showing dopamine is actually a very strong Th2 inducer for naive T cells. So what these publications have shown is dopamine can induce IL-13 production from naive CD4 T cells. And we confirmed that this is we isolated naive CD4 T cells, and then we put it in culture without any stimulation. So these are Th0 culture. But if you add dopamine to this Th0 culture, we see a significant increase in IL-13 mRNA levels. But if you treat the same Th0 culture with epinephrine or non-epinephrine, this is the adrenergic nerve marker, we didn't see the induction of IL-13. So that really shows the dopamine in the early life may have a very different function from epinephrine and the non-epinephrine in the later life. So although these two papers, as I mentioned, has already shown that dopamine can induce the Th2 phenotypes from naive T cells, they did not narrow down the mechanism. So they did not know what the receptors are in the T cells that mediate this response, and most importantly, how the intracellular signaling pathways are for regulating IL-13 production. So what we did is we wanted to take a look. So what we did is we look at all 5 dopamine receptors. There are only 5 dopamine receptors. Dopamine receptor 1 and the dopamine receptor 5 belong to D1 type. So this is the receptor that activates cyclin-MP production. And then D2, D3, and the D4, this is D2-like dopamine receptor. This is an inhibitory G-alpha-I induced G protein coupled receptor. The outcome is a reduction of the cyclin-MP intracellularly. So when we analyzed all these 5 dopamine receptors in T cells, as well as innate lymphoid cells, ILCs, what we found here is the DRD4 is the major receptor expressed by T cells. We did not find DRD4 in innate lymphoid cells. I know there's a new publication published in Immunity last month showing DRD1 they found the DRD1 in the innate lymphoid cells. We couldn't quite reconcile the differences. We did exactly the same culture, we did exactly the same condition, but in our hand, we cannot confirm the DRD1 is expressed by innate immunity cells. So that issue still needs to be sorted out. So, and then after we confirmed at the mRNA level, DRD4 is expressed by T cells, we look at the protein level. So protein level staining here shows very clearly the isolated CD4 positive T cells, they all have the surface expression of DRD4. The mast cells do not have DRD4. We also showed the dendritic cells, macrophages, also do not have DRD4. So DRD4 seems to be very specific for CD4 positive T cells. To confirm that, we look at the lung. So at the baseline, without any allergen exposure or challenges, there's very few T cells, CD4 T cells in the naive mouse lung. So we didn't find that much DRD4 staining at all. But if you challenge the animal with ovalbumin, which is one of the models we have to induce allergic inflammation, we start to see DRD4 staining in cells that surrounded the airway as well as surrounded the blood vessel. This is where the T cells are being recruited to the lung. We've validated the antibody is highly specific by using the DRD4 knockout animal. So you can see there's no staining of DRD4 in DRD4 knockout animal, even with the allergen challenge. And then most importantly, the DRD4 positive cells, which you can show here, red, in this ovalbumin treated animal is in close proximity to Th positive nerves. So this creates a possibility. The nerve can release dopamine, and then dopamine can signal to DRD4 to regulate their Th2 phenotypes. In humans, we also validated DRD4 is very specific for T cells. So this is a human lymph node, lung-associated lymph node cross-section. As you can appreciate, where there is a high level of DRD4 expression, there is also a high level of CD4 expression. So with all these evidences, we were pretty confident DRD4 is very specific for CD4 T cells. To further confirm DRD4 not only bind to the dopamine, also DRD4 mediates the IL-13 induction, we did the following experiment. The first experiment is, as I showed before, dopamine can induce IL-13 production from TH0 cells. These are the human TH0 cells we isolate from the lung-associated lymph nodes. The dopamine effect can be reproduced by DRD4 agonist. So A412997 is a specific DRD4 agonist. The agonist creates the same effect as dopamine in this culture condition. And then this dopamine effect can be completely reversed by DRD4 antagonist. Same phenotypes were seen in mouse CD4 cells. And then more importantly, if you isolate the CD4 cells from DRD4 knockout animal, there's no more effect of dopamine on IL-13 production. So with this evidence, we concluded DRD4 not only is selectively expressed by T cells, DRD4 is also the functional receptor to activate IL-13 upon dopamine stimulation. So then the most critical question here is, does dopamine play a role? Dopamine-induced IL-13 play a role in allergic phenotypes. So what we did here is we inhibit DRD4 with the inhibitor I showed you previously. So this is a L745-870. This is a DRD4 specific inhibitor. So what we did here is we used this neonatal animal model. And then after the sensitization, we started to treat with DRD4 inhibitor. And then following the challenges, we measured the type 2 information. We did a whole panel of type 2 cytokines, chemokines in the lung, in the BAL. We also did the mucus as well as airway reactivity. Because the time limit and also because the paper has already been published, I'm going to just show you the type 2 cytokines in this figure. So what you can see here is how dust mite exposure induces type 2 cytokine production, shown by here, the IL-5, IL-13, and then there's much more lymphocyte recruited to the lung. Similarly, eosinophil number is also increased. But if you treated the animal with this DRD4 specific inhibitor, you completely blocked these allergic phenotypes shown by the normalized IL-5 levels, IL-13 levels, as well as much significantly reduced eosinophils and the lymphocytes. So this really shows the DRD4 pathway is very important for the initial Th2 response in this neonatal animal model. In contrast, if you start to do the same experiment in the adult model, in this adult model, everything is the same except the sensitization challenge is with 3-month-old adult animal model. What we found here is the DRD4 inhibitor no longer have any effect on Th2 response. So this really is consistent with the idea there's a rate-limiting ligand in the lung. So we showed previously dopamine level is much higher in the neonatal lung. As a result, the DRD4 signaling is much more active. However, in the adult lung, the dopaminergic nerves becomes adrenergic nerve, and then this effect of dopamine no longer operate in the adult system. So to confirm that, we actually can isolate the adult T cells. So what we can show here is the DRD4 expression is actually not affected by age, independent of whether you are using Th0 condition or Th2 condition. And if you just treat with adult T cells with dopamine in vitro, the dopamine effect is similar between postnatal day 21 T cells and adult T cells. So this really gives you the idea it's truly the environment in the lung that modulates the susceptibility to allergic inflammation, is not T cells. So to summarize this part of our talk, as I showed you, in the early development in lung, what we believe here is there's a much more robust, much more abundant dopamine in the lung because the nerve at this time is dopaminergic. And then this dopamine can signal to DRD4 receptor and activates 2 different pathways we showed in this paper. One is to activate IL-2 receptor alpha expression. These can activate the Th2 differentiation. The other pathway is to inhibit EZH2 PRC2 complex. So this is a epigenetic modulator that can repress IL-13 locus. So through these 2 pathways, dopamine that's secreted abundantly in the neonatal lung can promote Th2 differentiation, therefore exacerbate the initial allergic response. However, in the adult lung, although T cells express the DRD4 receptor, However, the dopamine level is much reduced in the adult lung. As a result, this pathway no longer operates. So this is here, I summarized here. Basically, what we are adding to this whole entire picture is, in addition to IL-2 and IL-4, that's can be produced by T cells themselves, there are additional environmental signals, such as dopamine released by the developing nervous system that can regulate how naive T cells become Th2 cells. The dopamine can facilitate this process. As a result, the developmental lung environment is actually much favor of Th2 phenotypes. Maybe that explains why the initial age onset of allergic asthma is usually in the early childhood. However, this is clearly not the complete story. As I said, asthma is a progressive disease. What happened is once you have an initial Th2 differentiation, these Th2 cells will, most of them will die if you do not have continuous stimuli. However, the few Th2 cells that remained in the lung that do not die after initial allergen challenge will become memory Th2 cells. These are the cells that reside in the lung. Recent studies shows the memory Th2 cells are the key mediator of recurrent allergic inflammation. So what happened is these are the cells already poised. Once you have a second challenge using the same allergen, these T cells will get activated very, very fast and very quickly. So this is the true problem for the chronic asthma patients. So with that question in our mind about this contraction phase, because I showed you the dopamine is high up to here, P21, and then we measured in adults, dopamine level is low. But what happened to the contraction phase? So before we do that, we wanted to build up a system where we can actually isolate and track the Th2 resident memory cells. So if you look at the literature, there are 3 different types of memory cells: central memory, effector memory, as well as resident memory. So in addition to all these different markers you can use to sort different type of memory cells, we'll also use another criteria to make sure the cell types we're isolating are truly memory cells. So this is just the cartoon showing you in the lung. The central memory in red, the central memory is in green here. Central memory cells is usually in the lymph nodes. And then effector memory cells is in blue. They can be in the lung. But the effector memory and the central memory cells, they all go through the circulation. So they constantly circulate. And then they have to go through the circulation to get back to the lung. But the resident memory cells, they do not go into the circulation. So what you can do for distinguishing the central and effector memory cells from resident memory cells is to do an intravascular CD4 labeling. So what we did is whenever we isolate TRMs, what we use is these very specific markers. In addition to that, we use an intravascular CD4 negative marker to distinguish the T cells in the circulation from T cells residing in the lung. So from now on, if I talk about Th2 TRMs, these are Th2 cells that express CD44 high, CD69 positive, CD62 negative, and then circulating labeling negative. So this is the very stringent definition of resident memory cells in the lung. So let's focus on this phase. This is the contraction phase. When the Th2 effector cells, they have to die. The majority of them die because they lose the continuous stimulation. And then eventually they become memory cells. So this becomes the time window when the memory cell formation can be modulated. So following our initial discovery, the dopamine level is high in neonatal lung, but low in the adult lung. We decided to look at what the dopamine innovation looks like during this two-week transition time window. So what we found here is the dopamine level actually maintained relatively high during this contraction phase. In addition to this evidence, we also did a systematic screen for all kinds of neurotransmitters that can regulate IL-13 production from already differentiated Th2 cells. Just remember, resident memory cells come from already differentiated Th2 cells. So in this particular experiment, we differentiated naive T cells to Th2 cells, and then we stimulated Th2 cells with different neurotransmitter to see which neurotransmitter can further exacerbate the Th2 phenotype. we're using IL-13 mRNA as a reporter. So if you look at all these neurotransmitters, these neurotransmitters are all known to be present in the lung. This is a dopamine. So dopamine clearly stands above beyond any other neurotransmitters. The other neurotransmitter that can induce IL-13 production is substance P. This finding has already been published by another MGH group published in Neuron in 2015. So to some extent, our finding in substance P can potentiate IL-13 production in Th2 cells, validated already the previous finding. However, compared to the substance P, dopamine have a much stronger and much potent effect on the IL-13 production. So then we decided to run RNA sequencing to look at what dopamine is doing to already differentiate Th2 cells. So what we found, again, confirmed with our screen, is IL-13 can be significantly induced by dopamine in Th2 cells. IL-2 expression is also upregulated. IL-4 is also upregulated. IL-5. So all the cytokines that are known to regulate Th2 information can be enhanced by dopamine in already differentiated Th2 cells. And very interestingly, the genes that are known to promote the aggression of T cells is all downregulated. For example, S1P1R. So this is the receptor you have to block to keep the cells in the lung. So anyhow, so all these findings consistent with the idea that dopamine actually facilitate Th2 phenotype at the same time promotes the resident phenotype. So these are the pathway analysis. A few pathways that are known to regulate residency is TGF-beta, IL-2. This is all elevated, but the pathways that regulate cell migration, as well as the integrin signaling, which are known to regulate the aggression of the lymphocyte, are downregulated. So again, this is consistent with the idea dopamine can change the Th2 TRM phenotypes in this assay. So since we have the gene signature regulated by dopamine, we decided to take a look at what other resident memory cells look like. Whether any of the genes that can be regulated by dopamine has also been seen to be enriched in resident memory cells. So we decided just to look at the literature because there are many publications already describing CD4 and the CD8 resident memory cells in different organs. So here is the spleen as a control for central and effector memory cells. And then here is the 3 different organs people already published for the resident memory cells. So what we believe here is the residency program likely is regulated by a similar set of genes. So if that's our belief, actually it's not just our belief, there is many other publications suggesting the central regulator of any tissue residency is likely shared between different tissues and between different T cells. So here is the CD4 T cells and CD8 T cells. So what we found here is there are a core TRM genes similarly regulated in CD4 positive TRMs and the CD8 positive TRMs. So these are the genes, okay? And then all the genes that are differentially regulated by resident memory cells, independent T cells, are similarly regulated by dopamine. You can see that this is a dopamine treatment. The dopamine downregulate genes that are known to be downregulated in CD4 or CD8 resident memory cells compared to the central and effector memory cells. And the genes that are upregulated is also similarly upregulated in the resident memory cells across different tissues. So this is basically the bioinformatic calculation. So after all this calculation, about a third of the core resident memory genes are similarly regulated by dopamine. So this will give us a lot more confidence. Maybe dopamine play a role in promoting the residency of already formed Th2 cells. So, so is that true? So we decided to look a little bit harder using lineage tracing. So we wanted to look at where the Th2 TRMs are in the lung, especially in relationship to the dopaminergic nerves. So we took advantage of the IL-13 Cree system. So this is IL-13 mouse. In the baseline, there's so few IL-13 cells, so we cannot find any IL-13 lineage labeled cells. But if you give the animal allergy, in this case it's house dust mite, very quickly after 2 days, this is acute phase, you have a lot of red cells appeared around the airway. This is the blood vessel. You can see there's a lot of red cells. If you waited for 10 days, So this is the time window when the resident memory cells start to form. You can see there's a significant reduction of the number of lineage labeled cells. So precisely as literature told us, the contraction phase is when the Th2 cells, majority of the cells die. We estimated about 80% Th2 effector cells died during this contraction phase. Only about 20% IL-13 producing cells are maintained in the lung. So if you look at the nerves are labeled here by green. So, and if you overlay, what we found here is comparing 2 days versus 10 days. 10 days, majority of the labeled Th2 cells actually are close to the nerves. Here is a zoom in. You can see the green is the nerve, the red is Th2 cells. And then, but in the acute phase, although there are a lot more Th2 cells, the number of Th2 cells that are closely related to the nerves is actually much lower. So in another word, we see an enrichment of Th2 TRMs, resident memory cells, clustering around the dopaminergic nerves. So this truly gives the opportunity for the dopaminergic nerves to interact, to guide the Th2 cells, maybe to take on a resident memory phenotype. So here is just to give you the summary. What we showed here is during this contraction phase, we're using lineage labeling to label the Th2 cells. And we found the Th2 cells, although the number of Th2 cells drastically decreased during this contraction phase, the proportion of Th2 cells that are lineage labeled that are close to the nerves actually significantly increased. So we then speculate if dopamine can produce Th2, promotes Th2-like phenotypes, and then can promote resident memory residency, can it contribute to the memory Th2 phenotypes? So to do that, we have to rebuild the whole system. As I said, all the memory cells are studied in the adult lung. So the first thing we did is to validate whether the neonatal model can similarly induce a memory phenotype. So to do that, what we have to do is the second challenge. So this second challenge is really activating the memory cells because there's no more sensitization. So sensitization happens here. This is the effector cells start to form. And then at this time window, you should have memory cells if the neonatal system does allow the memory cells to form. And then if you challenge again, you will have a very, very robust phenotype. And then turn out to be true. So here we have 4 groups of animals. Everybody is sensitized. But there are a group of mice that only get one challenge, and then there are groups that get both challenges. As you can see from different parameters, only the mice that get both challenges have a much significant allergic phenotype. Anybody who gets only one challenge only has modest or not any significant allergic information after all. For this particular challenge at the neonates, after 5 weeks, the allergic information is already largely dissolved. So with this evidence, we believe the neonatal model also support a memory program in the lung. So to be thorough, what we did again is we used an inhibitor. So this is a FTY720. This is a standard inhibitor to promote to block the aggression of the immune cells. So in another word, if you treat an animal with FTY720, all the all the T cells that are in the lymphoid cannot come to the lung because they cannot egress from these lymph nodes. So whatever the cells that mediate this allergic phenotype has to do with the resident memory cells. So in this case, I'm going to walk you through. So now there are 3 groups of animals. There is no challenge, which is the baseline. And then we have double challenged animal I showed you before. This animal mounts a very, very robust allergic phenotype. Among these double challenged animal, one group is just treated with saline, another group is treated with this blocker of aggression of immune cells. So this blocker will allow the immune cells only present in the lung to act. There's no contribution from circulating T cells. So what you can see here is even if you block the immune cells coming to the lung, the allergic information is as robust as no block at all. So if you see this data, you draw the conclusion that immune response in this double challenge model is mediated predominantly by the immune cells in the lung. So basically, we have all the parameters. These are just the major parameters I'm showing you today. Is MUC5AC independent whether you block the aggression of the immune cells or not? They are the same. And most importantly, the airway reactivity is also similarly mediated only by only by the resident memory cells, because the blocker doesn't reduce this airway hyperreactivity. So with that, with all this basically builds the model. The neonatal model not only allows the memory cells to form, the memory cells that formed from neonatal allergen exposure also are sufficient to mount the recurrent allergic response. So now we have this neonatal model set to go. So then we did the second layer of the experiment, which is using this neonatal model, which now we know has a resident memory program. We inhibit DRD4 only after this first challenge. So remember, this is the time window when the Th2 cells are already formed. They are going to die out to become the memory cells. So instead of blocking the acute inflammation, now we block DRD4 signaling during this critical contraction time window. And then we analyzed whether there is a change in the memory cell number. We found inhibiting the DRD4 during this contraction phase does it reduce the memory cell number in the lung. And then to provide functional evidence, in this case, we did two combinations. So one is with the DRD4 inhibitor during this contraction phase. Another is to block the aggression of central memory and the effector memory cells, so that the effector memory cells in the lung at this rechallenge condition will be the resident memory cells. So as I showed you, I already showed you, if you have the DRD4 inhibitor, you get reduced Th2 resident memory cells. And then again, consistent with the FTY720, only block the circulating memory cells. Similarly, if you just block in the circulating memory cells, there is no change in the resident memory cell number. If you use the both combination, you similarly reduce the memory cells as just use the DRD4 inhibitor. So here is the allergen challenge model. This is the functional model showing if you block the DRD4 signaling, you reduce the airway reactivity. So basically this finding shows not only you reduce the number of the resident memory cells, you also reduce the activity of the memory cells with the DRD4 inhibitor. So here is basically the conclusion. What we found here is the dopamine during the contraction phase signal to DRD4 and then can regulate the development of the TRM, the resident memory cells. And then not only reduce the number, also reduce the function. As a result, if you block DRD4 signaling during this contraction phase, you can alleviate the recurrent allergic inflammation. So I can stop here. So to take questions, or I can keep going, but I think here probably is a good time to stop. So I guess ultimately I will need to thank the team. I want to thank, as that Wei I already introduced. He is currently an instructor at MGH. Yan is going to be promoted as assistant professor. She's a KO8 awardee. She's the one who studies the airway smooth muscle. And then I have all these wonderful people worked previously with me. Caroline is now assistant professor at the Bay State Hospital. All my students, Lynn, Cody, Juliana, they all go on to become principal investigators in company. So all of them are currently hold the principal investigator positions in the company. Jonathan is UC at the MSTP student in UNC. And then I want to thank funding. The CARE Award is for Dr. Yan Bai, who is a pulmonary fellow at MGH. So I can take questions now if anybody has more questions.